with different individuals run against it, and you can compare to see whether they have any bands that don't belong to one of the parents. So in courting males during courtship time, they'll sing while they display their hackles. So singing isn't one thing that I've looked at yet, but it's going to be very important in mate choice, I think. But these hackles we've concentrated on right now. With respect to singing though, the latest studies were done in the mid-1990s by Mountjoy and Lemon, and they found that first it was known that starlings are open-ended singers, so as they age, their songs become more complex. They sing more song types, they have a larger repertoire size. And so females might be able to judge or assess males with respect to how old they are by their song. If they're singing really amazingly long songs or really intricate songs, then they're likely to be older. So they had, in Mountjoy and Lemon, looked at this, and they found that males were in better condition, the ones that sang more songs or song types, and they paired faster. They didn't look at genetic success or anything like that, but they found that they were selected quicker as mates. So what we did is we looked at the hackles, and we found that males had significantly longer hackles than females, and it's not surprising, and that they also had longer iridescent portions to their feathers. So this just shows that the males, median is about 10 millimeters of iridescence, and the females just around 6. Now, this is interesting. What we do when you study a population over a period of years, especially banded ones, you can start to see patterns. And these are females that have been caught in more than 1 year. I know it looks like a lot of numbers, so I'll go through it with you. But let's just grab, if you look here, this is the iridescence length. So this is a female. This is a female. So each, after a space, that's a different female. You can see that iridescence length typically increases over time. So this female, number 7, was caught in 2007 as a young because her hackle length was only 4.4, quite a bit under 6.5, and then that. So you can also see their site fidelity to nest boxes as well, not to mates, but to different boxes, and the males will show this as well. So this is just, again, another one. This female was actually in 2011, she was one of our first ones that we caught. In 2011, in April, we found her dead in the nest box, but she had incredible reproductive success. Now these are males, same thing for males. You can see that over time, these iridescence lengths keep increasing. The total hackle length, not always, okay? It's not as reliable as the iridescent length. So the total length of the hackle, so the full length of it, increased more reliably with age in males than in females. But the iridescent length of adult male and female hackles seem to lengthen with age. So we also found that those lengths were associated with conditions. So adults with longer iridescent portions to their feathers were older and in better condition. So they were in better condition. Now this makes sense. We found that there were a lot more young males breeding in 2007 than in 2011. Because when we put the nest boxes up, we started in November and ended in February. So I've seen just today, starlings are going around scouting for places to nest for next spring. So we think the older ones have already found their spots. And so suddenly there's all these new nest box opportunities that the young ones were able to come and breed in. So we found 85% of young males in 2007 versus 48% in 2011. Here for females, 73% versus 32%. So we're seeing a lot of returning starlings coming back, and of course they're going to be older, and they seem to stay in the boxes despite what my students and I do to them. So. Now, one of the things that we found, and I just kind of jump into this, is we found within pair young. So within pair young, and that's a lot of numbers to look at, but within pair young are nestlings that are the product of the mom and dad, the social mom and dad. Extra pair young are genetically related to the mom, but not the dad. Now we get interesting. We found quasi-parasitic young, and those are nestlings that belong to the dad, but not the mom. So how does that happen? How do you get a nestling in there that belongs to the dad and not the mom? So that male has gone out and copulated with an extra pair female, who then comes and lays her egg in his nest. So we have, it's pretty steady over the years, and but you can see that anywhere between 31% to 45% of nests had some quasi-parasitic young in them. And they accounted for about 15% of the young. And then there's intrapacific brood parasitic young, and that just means that a female from nest X will lay her egg in female from nest Y. And it's not related, so you've got a nestling that's not related to the mom or the dad. So we've got quite a system going on here. It's really complicated when you're trying to assign paternity, because it's not just looking for who the dad is, but you're also looking for the mom, and it gets interesting. But you can see that this is going to allow us to ask a lot of different questions. So of all the young, on 62 broods, we found that only 6, most of these broods are mixed. So there's mixed paternity, mixed maternity. Only about 10% contained only within pair young. Maybe 5% contained only extra pair young, 5% only the QPY, and 3% contained only IBPY. So basically there's a mixture of different maternity and paternity in those other nests. Now, what we found, because we had funding back then to do DNA fingerprinting, older adults produce more genetic offspring. That's interesting. You would expect that older ones are in the bird literature are often seen as higher quality. They've got experience and they've proven that they're good survivors. They probably have good genes, you could stretch to that, so that they're going to sire more genetic offspring. That just means offspring in their own nest and offspring in other nests as well. So males had on average 3.4, a range of 0 to 10. We had some super males in there. And females 3.3 with a range of 0 to 8. So if you look at here, you can see the young versus the old, and the old is quite a bit higher. The thing that's amazing about this is this, how equal the males and females are. Usually what you expect to see is most of the variation in reproductive success is by males, because they're the ones that can go out and mate with as many willing females as possible. The females are constrained by the number of eggs they can lay, but these females aren't. Because they are able to lay eggs in other conspecific, other starling nests, it's thought that they are indeterminate layers, so that they're able to keep laying more than, say, the average songbird. So it's amazing that they're able to produce as many as males. So if we look just here, it's the presence of genetic offspring. 43% of females, I just want you to notice how similar these numbers are across the board for females and males. So if they had young in their own nest and had also sired them in other nests, only in their own nest, only in other nests, and no genetic offspring at all. So there's some unlucky adults out there that, despite all their hard work at provisioning, didn't gain any maternity or paternity. But we found that, we thought maybe older males and females are going to be engaged more in extra pair copulations, but we didn't find that age had any effect on predicting who had within pair young, extra pair young, that sort of thing. Nor did it predict which, we thought maybe older females would get more young in other nests through parasitism, no. And older males didn't sire more extra pair young in that way. But we did find that older, remember we said that older adults sired more genetic offspring, so it's got to be coming from somewhere. They produced more within their own nest. So they had more paternity or maternity within their own nest, which raised them up to be higher levels of offspring. So the old 2.94 versus 2.11, that's males and females combined. So they did not produce more genetic offspring in other starlings' nests. So where they're getting their increased reproductive success, more genetic offspring in their own nest. We also found that they chose each other. It so happened, I mean, we can't show it because it's correlational, but males with long iridescent components to their hackles chose females with long iridescent components to their hackles, both as social mates and as sexual mates. So there's a sort of mate choice going on. So I think we can say we need to do some manipulations, but it looks like hackles do signal age and condition in both sexes. So if it's condition dependent, so it's honest, because if you're not in good condition, you can't have, or you're not older, you can't have these longer hackles. It's something that comes with time. It's probably really useful for mutual mate choice. So it's showing here, because there hasn't been a lot of work done on how males choose their mates. It's usually done how females choose their mates. But with this species, you're able to look at both sides of the coin. So the next thing we did was we investigated the relationship between the color of those hackles, that beautiful iridescence, took them into a lab in University of Windsor and put a spectrophotometer on them and quantified the hue, the chroma or the saturation, and the brightness of those feathers. So that was done by my former master's student, Joel Slade, who's now at Western doing a PhD. So we looked to see, we saw there was a study out in an aviary, of birds in an aviary where female starlings preferred to spend time with purple feathered males more than the green feathered males. So we thought, I wonder if that translates into actual mate choice in the wild. Will those purple males sire more offspring? And are they better fathers? Are they in better condition? Now birds see in ultraviolet light. So humans can see from about 400 to 700 nanometers, whereas birds can see in the UV spectrum, which is about 320 to 400. So they see things that we don't. And so the plumage that we see is different than what they see. So when you're doing this sort of study, it has to be under conditions as the birds would see it themselves. So we did find some interesting results, and that was that different parts of the coloration signaled aspects differently between the sexes. So I didn't explain that very well. But age, for males and females, the color of the hackles predicted how old these birds were. With males, it was the brightness and the hue. So think of hue as whether it's purple or green. With females, it was the brightness of those feathers that showed how old. So older females had brighter feathers. With males, bright and more purple, they were older. Condition, bright feathers also showed that they were in good condition. And males with really deeply purple feathers, they were very good providers to the offspring. With reproductive success, in females it was hue. So the very purple feathered females, they had highest reproductive success. But you didn't see that for males. So the coloration is giving different information to members of the opposite sex. So just some things to show, it says it better. Hackle color communicates different information in males and females. So here, I've just got something showing colors. This is highly bright females versus not very bright females, and this is plumage-wise, with respect to body condition. So you can see that very bright females were in better condition than very not bright females. Here, this is males, and this is just a principal component analysis, and it's just holding a lot of variables constant at one time. Here you have high UV saturation and low UV saturation, and what's this for? So this is provisioning, parental effort. So you can see males that have high purple UV saturation are much better providers than ones that don't. And here we've just got reproductive success. So females with UV green plumage didn't do as well as females with UV purple plumage. So do you have questions about that at all? It's a lot to take. Okay. So the last thing that I'm just going to talk about before I talk a little bit about what I might do in the future, I had a couple students really interested in ectoparasites of birds. And this was Mark. He's finished up his master's, and Evan is at Dalhousie doing his PhD. I think he might be finishing now. But what we were finding were, this is what starling eggs look like when they're first laid. And then some of them, especially in the second broods, I should say they lay 2 broods a year, they would get these spots on them. And these spots, we did our research, it looks like it's likely blood. One of the different parasites that are common in the nest are these things called carnis. They're very tiny, they go incredibly fast, and their preferred habitat are unfeathered nestlings. So they're blood feeding, so they're hematophages, they like to suck the blood, that's how they get to their next life stage. And too many of them can decrease condition in nestlings. You can imagine having a blood drainage. Some scientists in Europe had studied the spotless starlings, and they found that these were from the ectoparasite carnis. And so what they are, it's either the carnis after it's being on the adult, and then the adult applying the brood patch to the eggs, it might be squishing them, or else it's fecal matter with blood in it. The sister species, spotless starling, they found that it was tied to, so more spotted eggs were high infestations of this ectoparasite in the nest, and they found that it acted as a signal to the parents. If the male looked in and was incubating eggs that looked like this, he fed at a higher rate than males who had heavily spotted eggs. So we saw that, oh, isn't that odd? I would have thought that if you knew your kids were handicapped or they had something going against them, you'd maybe want to up your provisioning to get them back up there. Because if they're having, their resources drained away by these blood-sucking ectoparasites, maybe you should just up your feeding rates to help them out. So we looked at this, and we found that it was that the level of spottiness was not tied to the number of carnis in the nest, which was unusual. So we don't know where these spots are coming from in our population. But we did find that the adult males upped their feeding rates to eggs that were heavily spotted, to those nestlings, which is interesting. So what's next? I've just finished about 8 weeks of intensive thought about this. I put together a grant, sent it off to NSERC a couple days ago. Stephen Harper's government will be looking it over and seeing that there's absolutely no benefit, there's no profit, there's no foreseeable benefit of this research, so it probably won't get funded. But if you don't try, you'll never know. So I thought that the next steps I'd love if I had some money, would be to manipulate the iridescence length. So maybe cut some of that iridescence off and see how it affects. I was thinking I'd love to make some longer, but you can't really glue those fine little feathers. So I thought about that for about an hour and then thought, I don't think so. I forgot, and hue. Remember how we said the females that were most purple, that they had more genetic offspring? So it would be kind of neat, and maybe just to take a Sharpie and take away some of that. I can't make it more purple or more green, but I could sort of diminish that purple hue and see the effects. because by doing those kind of little studies, it actually can tease apart what you think is core, what's